Welcome back to Professor Enriquez's science lectures, for lack of better branding. I guess I should brand it. Hashtag something. Whatever. This is chemistry lecture part two. And as you can see, I'm going to be talking about the macromolecules. I've already covered in video part one the basics of how bonds are formed and the mechanism by which the electrons are shared. And remember that electrons may be shared evenly or unevenly, and whether it's even or uneven makes a very big difference, as we're going to see fairly soon. There are four groups of macromolecules. The first group that I'm going to talk about are the carbohydrates. And the carbohydrates, some sample molecular structures are shown here, as you can see. And one of the key things I want to point out right off the top with carbohydrates is all of that oxygen. You see, as you can see, I've got this chain of carbons. All the macromolecules have a chain of carbons, all with these lines that all represent covalent bonds. So they're all covalently bonded to each other. And there's hydrogens, which all the macromolecules have hydrogens. So carbohydrates have something else, so they have oxygen. That's where they get their name from. Carbo, and then hydrate refers to OH in this case, or water. It can also mean water. So these OHs are very important, because the carbohydrates have those oxygens, and oxygens are electron hogs. They have a high amount of what's known as electronegativity. And electronegativity means that the oxygen pulls on the electrons strongly when it's covalently bonded to something else. Obviously, oxygen and oxygen, when they bond together with each other, are going to share evenly. But if you have oxygen and carbon bond, or oxygen and hydrogen, the oxygen kind of sucks up more of the electron density. So that means that carbohydrates are hydrophilic because they mix with water, because they have little positive and negative charges all over them. And remember from part one, water has a lot of positive and negative charges all over it, which allows it to form hydrogen bonds, which is why carbohydrates also form hydrogen bonds. Now, carbohydrates come in these little 5 or 6 ring sugars. This is an individual. Glucose and fructose are examples of sugars, which are individual molecules. Those are called monosaccharides, and if you put them into a long chain, then you can end up with something called a polysaccharide. This is here on the bottom left, is an example of a oligosaccharide or a polysaccharide that's kind of small. And I've got several glucose molecules, and they're all in a long chain. Each one's bonded to the next, right? And then on the lower right, we have a molecule of glycogen. There's a little protein in the middle, and then all around here, these are all glucose molecules, all in all these sugar rings, all in long chains. This is actually how our own body forms starch. This is actually the starch that's stored in our liver and our muscles to help us deliver energy on demand. If you actually go sprinting, if you decide to go and do a 100-yard dash, you're probably going to start using up glycogen that's in your liver on your muscles right away. So now starch, and I refer to starch, I do mean glycogen as a starch. I also mean that the starch that's in potatoes, rice, and bread, that's also carbohydrates. So those, and remember, carbohydrates are, whether it's a single molecule or a long chain, it's basically sugar. So when you eat white rice or white bread, it breaks down fairly quickly into sugar, which is why people with diabetes are told they're not allowed to eat large amounts of rice or such things. Potatoes, especially French fries, are supposed to be very unhealthy for them. It's not really the fact that it's fried or even salty. It's the matter that there's a lot of sugar. It breaks down quickly. Whole wheat bread, brown rice, hulled barley as opposed to pearl barley, those are the healthiest kinds of carbs because they break down slower. And our body needs carbs, no question. Actually, these molecules, obviously, if we didn't need carbs to live, we wouldn't store this glycogen molecule. This is the molecule stored in our liver that releases carbohydrates on demand. So, so we need the glucose. The thing is that too much of a good thing can be a bad thing, because carbohydrates are sticky. They're hydrophilic, and so if you have too much sugar in your blood, it can actually gum up your cells, gum up your tissues. It can make your blood more viscous, and that's why diabetes is often associated with heart disease, because your heart's having to pump, really work a lot to move that syrup through your blood. So it's not how we want that to work. Um, the more bonds there are between the glucose molecules, the more complex we say the carbohydrates are. And in particular, if we have here on this left side, we can see these glucose molecules are attached to each other through a whole lot of covalent bonds. So many that it doesn't break apart easily. This is cellulose. This is an example of cellulose, which is the carbohydrate that's found in grass. And grass doesn't really break down into food. We don't eat grass for food. You eat it for fiber. You eat it to help you pass, feces through our large intestine. But so cellulose is an example of a highly complex carbohydrate. Obviously, more complex carbohydrate doesn't automatically mean it's better for you because we don't extract any carbs from this. Now, cows do, right? But we don't. So cows have 4 stomachs and we only have 1. So that would be part of the difference there. So carbohydrates are one of the groups of macromolecules. The second group is lipids. Carbohydrates and lipids are very similar in that they both contain a lot of carbon and hydrogen, but whereas the carbohydrates have a lot of oxygen, lipids do not. And these hydrogens are covalently bonded to the carbon atoms. That's the only kind of covalent. That's the only bond we have here, covalent bonds. This is a nonpolar bond. And so the fact that the bond is nonpolar, the electrons are evenly shared between both of the atoms in this case. That means that this is going to be a nonpolar molecule, which means it's hydrophobic. And the reason, and that's why oil makes these beads, right? And also, if you ever see a lava lamp, that's a good example. That's basically wax. It doesn't mix with water because wax is also falls under the category of a fat. And the thing is that the water molecules are attracted to each other through hydrogen bonding, right? And if I added some sugar, the sugar would mix with the water as well. It would be in the clear substance here. But there's no positive and negative charges on this entire lipid molecule, except over here, these little guys here, the ester bonds in the middle. But we're setting those aside. This is a very large molecule, and it has no oxygens throughout most of these tails. So that means that there's no reason for water to be attracted to it. So the water molecules only stay with each other. They don't mix with the oil. The oil has no reason to mix with water. And so you end up with something that separates from water. And that's why it's called hydrophobic, meaning afraid of water. So there are 3 different types of lipids that we very commonly see in the human body and in living things in general. So triglycerides are the fat molecule we use to store energy. So that's the one that's stored in our adipose tissue under our skin and so forth. And there's 2 kinds, saturated and unsaturated fats. The difference is whether or not they have these double bonds. The saturated fats have no double bonds. Unsaturated fats do. And so these are energy storage molecules. They're obviously also useful for insulation and padding, protection. So your kidneys have a lot of fatty padding around them. But those are all triglycerides. And they're so called because they're 3 fatty acid tails attached to this guy. Here's called the glycerol molecule. That's why. That's a 1. This is energy storing lipid. Cholesterol is also a lipid, but it's not an energy storing lipid. Cholesterol, this is the cholesterol here up top, is used for a lot of chemical processes. It's used to make testosterone and estrogen. It's used to make bile salts. It's used to make vitamin D. It's important in our cell membrane functionality. So it has a lot of key functions, but it doesn't provide energy. We don't break it down for energy. We also, most of us, I don't know about everyone, but most of us have enzymes in our livers that can turn fatty acids into cholesterol. And so we tend not to have problems with lack of cholesterol. Us in America tend to have a problem with excess cholesterol and how much of that is due to diet versus exercise versus other factors is not clear. It seems like there's probably a lot of genetic factors involved. But if you have too much cholesterol in your system, and that cholesterol forms these chunks of hydrophobic, right? Because remember, they don't mix with water. So these floating chunks of fat can get stuck to your arteries and make them kind of rough and makes the water not flow evenly across it. Then you end up with heart problems, right? And arterial problems. The third molecule that's of importance here is the phospholipid. And the phospholipid is shown here. It's very similar to the triglyceride. It has 2 fatty acid tails, but the third tail, instead of being a tail, is a phosphate here. You can see this phosphate head. It's got a phosphorus. And notice the phosphorus and 1, 2, 3, 4 oxygens right around it. That's very negative. It's got a lot of high electron density. And then the carbons around it are very deprived of electrons, and so they're very positive. So that means this is a very polar region, and then all of this part here is nonpolar. And that's very important because phospholipids are used to create our cell membranes. The cell membrane is a structure that goes all the way around the outside of the cell, and you can think of it as this kind of very thin layer of oil, not a solid bubble, right? The cell is not a blob of oil. Instead, it's just got a very thin, oily boundary around the outside. And it does that by having these long tails, which are made of fat, a lipid, right? Those are oil. And then the phosphate heads are naturally attracted to the water that's around, it's both inside the cell and outside the cell. So you end up with this bilayer, 2 layers, right? And each one, the phosphate heads are facing outwards away from the center. And the center here is hydrophobic. And so this makes a border that goes around the cell. And this also means that anything that mixes with water, so salt, sugars, proteins, all of those things don't tend to cross the oil membrane. They have no reason to, because their oil membrane is where there's no charge, and then water has a lot of positive and negative charges. So you end up forming this barrier that most of the things inside your cell can't cross. A few things can, oxygen and carbon dioxide can diffuse through, but most things stay on one side of the membrane or on the other, and they don't cross. So let's think about this. I've got oils and I've got carbohydrates. One mixes with water and one doesn't. Question, if you get oil on your hands, how do you get it off? And usually the immediate answer is people say soap. And you're right, but that's not complete. It's not actually the most accurate way to think. Because think about it, if I get oil on my hands, and I say, oh, my hands are gross, they're full of oil, and I go and put some soap on it, now my hands are dirty and soapy. They're not clean. So what happens next is I run water over my hands and the oil is gone. And the reason that happens is because soap is what's called an emulsifying agent. The soap is like a detergent. What it does is it is also kind of like the phospholipid. It also is a molecule that has a hydrophilic and a hydrophobic component. And so what what soap does is it allows oil and water to mix. Going back a couple slides real quick, if I took at this, you see how I've got this fat, this looks like vegetable oil that's been poured into water, and it's created these beads, these separated out droplets. If I were to add some soap to this, then all of a sudden the boundaries would get fuzzy because the soap would allow the oil and the water to mix more. That's the function of soap. That's what it does. And that's what the word emulsify refers to. And we actually, when we eat fatty foods, we actually have a structure in our digestive system called the gallbladder, which stores a material called bile, which I just mentioned. It's made partially using cholesterol. And bile is like a soap. It allows oil and water to mix. So when you eat greasy foods, anything with fat in it, the more fat it is, the more bile you need released from your gallbladder to mix that oil with the water of your digestive system. And if you don't do that, if, for example, some people have gallbladder problems, then they end up not being able to digest all the fat they eat when they eat fatty foods, and then they end up getting the shits. So that would be a key component understanding lipids. So we've got 2 macromolecule groups down. We've got the carbohydrates and the lipids. Now we're going to cover amino acids. Amino acids are the individual molecules that combine together to make protein. And the human body uses 20 different kinds of amino acids. There's actually a lot of other ones too, but these is the 20. There's 20 that our human bodies use. And each one of them has a different they have the same structure in that they have a nitrogen with a hydrogen. This is called the amine group on one side. And then they have this carbon with 2 oxygens. This is called a carboxyl or a carboxylic acid group. So this is where they get its name. This is an amine and this is an acid. So this is an amino acid. Each of these amino acids has a different side chain in the middle of it, in the middle of the molecule. And so when you can make a peptide bond, which is the attachment between the amine group of one amino acid and the acid group of another amino acid, this is a peptide bond. It's a covalent bond. There's 2 electrons being shared between the nitrogen and the carbon. But what happens is that you can make a long chain of amino acids, but that chain of amino acids will be very different depending on which amino acids are in that chain. And an even slight difference in the amino acids, even one amino acid difference can change the shape of the protein. And so again, this is showing the peptide bond, showing the acid of one amino acid and the amine of another amino acid, and the bond, the peptide bond that forms between them. And then these different R groups may be a number of different structures. So, for example, glycine has a hydrogen, alanine has a methyl group, and then cysteine has this sulfur on here. And so each of these amino acids has a different property based on how they've been put into the chain. There's a diagram here showing all 20 amino acids. There's actually another one. I think it's got selenium in it. If your biology professor puts that on the test, they're an asshole. So these are the different amino acids. We need all 20 in our diet in order to be able to build new proteins. It's like trying to type with a keyboard with some keys missing. You can't make a good. You can get away with a few words, but you can't write a whole book with missing keys on a keyboard. So all of these different amino acids, they have different properties. And which ones you put on the chain is going to change the shape of the protein. Because what happens is amino acids, when they make this long chain, then they end up coiling up. They can make this spiral, or they can make these flat structures. And then the spirals and flat structures and all the different amino acids attach to each other. They kind of bond with each other with hydrogen bonds. Most of these amino acids are hydrophilic. There's some that are hydrophobic also. And they're going to end up creating a three-dimensional structure, which is going to be a protein. And proteins do most of the important jobs in our cells. Our enzymes are proteins, for example. Those are structures that make chemical reactions happen. The structures that actually give tensile strength, so toughness, hardness, well, not hardness necessarily, but flexibility, strength, resistance to stretching, those are all things that are done by proteins with collagen and elastin and so forth. There are structures that go into the cell membrane that allow some things through and not others. Like, for example, this might be something that allows glucose to move into the cell. Because remember, glucose can't cross the cell membrane. So if you have a channel like this, then glucose could pass through when it opens and not when it closes. And then insulin is a hormone that is created, is made of proteins, made of amino acids. So we've got lots of different kinds of things in our bodies. And every one of our cells, and between the cells also, because collagen is between the cells, and collagen is a protein that gives strength, toughness to our structures, like our ligaments and bones have a lot of collagen. So now here's the thing, that chain of amino acids, it has to be exact. You have to have the right amino acids in the right order. So where did that information come from? Well, it comes from our DNA. Our DNA is one of the types of the fourth group of macromolecules, which is nucleic acids. And DNA actually stands for deoxyribose nucleic acid. That's where the Na stands for. So that's the fourth group of macromolecules. DNA is an information storing macromolecule. It holds information in it by having the different letters A, C, T, and G. Those are molecules that we abbreviate them with those letters. The information that's stored in those letters A, C, T, and G, that is used to create the proteins. So notice that DNA has this long chain of carbohydrates with phosphate between them. This is a backbone. This is all covalently bonded. And then the two strands of DNA are attached to each other with hydrogen bonds. It's very weak. A hydrogen bond actually comes apart very easily. It's like a zipper. You can zip it and unzip it, so DNA closes and opens. And when it opens, then you can copy that information. That information is copied with an RNA molecule, and in RNA you use uracil instead of thymine for reasons that don't interest me. But this is how our information is stored, this DNA molecule. And the reason we use DNA for storing permanent information is DNA is the most stable of the nucleic acids. RNA breaks down more quickly than DNA. DNA can last a long time. And that's kind of the point of it. We keep a permanent copy of our information in our cell's nucleus, and when you need that information, you can copy with RNA and make amino acid chains, which are proteins, which again, as I mentioned, do things like enzymes are, again, made of amino acids, amino proteins. Structural things, like things that hold the cell together, all made with protein. And because enzymes and cytoskeletal structures and everything, they're kind of taking on a lot of wear and tear over time as they work to keep our cells working. That means they break down and they have to be replaced. So the ability to make new structures to replace old broken structures in our cells is one of the key functions, is why having a nucleus with DNA is so important in order for all of our cells to remain alive as long as they can. So one last thing on the DNA, this entire structure here is called a nucleotide. It's got the letter, right? It's a nitrogenous base. This is adenine in this case. There's other ones also. And then it's attached to a 5-carbon sugar called a ribose. And then the ribose normally has, when it's floating free like this is 3 phosphate groups. This is a molecule called adenosine triphosphate, or ATP. You may have heard of that before because ATP is also used by the cell to use energy. And when I say use energy, I mean that energy is delivered to the cell by having glucose molecules delivered to it. The cell will break down the glucose, in turn producing carbon dioxide, which we later breathe out. And then that energy is then trapped in ATP. It's kind of, ATP is the molecule that can actually bind to your enzymes and break that phosphate bond, right? The bond between the second and third phosphate. When that bond breaks, it releases the extra energy from it. And by releasing that extra energy, what happens is it allows that energy to go into making things like putting amino acids. Because putting amino acids into a chain is not free, right? Just going back here, this does not happen by accident. None of this happens by accident. You need energy to do this. You need energy to remove the water and put things together in the right order. So that means that ATP is the way that's done. And so the ATP molecules are constantly having their phosphate come off in order to put amino acid chains together. Then you got to reattach a new phosphate, in so doing putting energy into the ATP and taking it out of the glucose molecule. And that's what ATP is. Okay, so hopefully that all made sense. That's the end of the chemistry lecture, the 2-part chemistry lecture series. If you want more information on chemistry, I strongly recommend taking it from an actual chemistry professor. This is all the chemistry you would need to do well in a biology class. So hopefully that makes sense. And let me know in the comments if you have any questions or requests for other things. Obviously, I have a limited time since I am also a full-time professor, but I'll see what I can do. Thank you. And they create... Quincy, could you not, please?